Thank you very much. I've really been excited by what I've heard so far, and I hope I can contribute something along the lines of giving you a different perspective of how to think about stress, talking a little bit more about brain plasticity, which is something that certainly the first panel referred to quite a bit, and then giving you some thoughts and some examples, some of which you probably already know, of interventions that may be useful. So you'll notice that the word resilience has been used, and resilience I think of as the ability to achieve a successful outcome in the face of adversity. And we've heard that referenced a lot in terms of the brain. We'll talk about what that means, but one of the most important things to keep in mind is that gene expression in the brain, and the brain is continually changing as we go through our lives. So there's no such thing as reversibility. We can talk about recovery, resilience, and so forth, but we can't really talk about the reversibility of any of the things that are going on. So we're thinking of trajectories of our life course and changing those trajectories. So we all experience stress, don't fully understand what it is or how our bodies defend against us. Often we blame bad things on cortisol without realizing that we wouldn't be alive if we didn't have cortisol around to help us adapt. So how does this stress get under our skin, and what does it do to our brains and our bodies? So that's the first question. If we talk about stressors, the word stress, which we bandy about almost every day of our lives, we can talk about positive stress, to feel exhilarated from taking an exam successfully, giving a talk, things like that. To do this, we should have a good sense of mastery and control, a good self-esteem, and generally feel elated, or if something doesn't go well, then be able to show resilience and move on. Tolerable stress is something that happens to people who have good self-esteem, good sense of mastery and control, something like losing a job or ending a relationship, things of that sort, which are much more serious, but with good social and emotional support, and often material support as well, one can weather the storm and be resilient and move on. And then there's something that's in the title of the talk that almost speaks for itself. Toxic stress is when maybe the same things happen, but there is the person does not have those capacities to show the resilience, and this may actually then reflect a history of early life abuse, neglect, and so, and as we've already heard quite a bit about. So, but the other things that we have to bear in mind when we talk about stress is that there are other experiences that we don't use the word stress for necessarily, that are very much involved in our physiology that can cause us problems. And these include, of course, health-damaging behaviors, which often result from being under pressure, being under stress. They involve loneliness. They can involve circadian disruption, as in jet lag or shift work, or sleep deprivation for a variety of reasons, either self-imposed or imposed by one's lifestyle, and living in an ugly, noisy, polluted neighborhood with lack of green space. There's documentation that all of these things can get under the skin and disrupt physiology and affect the brain. And so we come to the brain as the central organ of perceiving what is stressful, potentially stressful, deciding, being vigilant, determining the behavioral and physiologic responses, and the behavioral responses may include those health damaging behaviors that I mentioned, being isolated and so forth, patterns of sleep and waking, and then the physiologic responses, which are influenced by those behaviors, but also directly output from the brain, involving the autonomic, the immune, neuroendocrine, and metabolic systems, normally lead to adaptation and survival. Hence, we wouldn't live without our cortisol or many of these other mediators, in a process that has been termed allostasis, meaning the active process of putting out these mediators in order to adapt and survive. But then several decades ago, we and others coined the term allostatic load, which refers to the price that the body pays for doing its job, especially when there is a lot of these incidences that force the system, a lot of stress, but also when the balance of the regulatory systems is dysregulated, and either you don't turn on things when you need them or turn them off when you don't need them. And so the metaphor for allostatic load is two heavy weights on the ends of a seesaw. The seesaw, or homeostasis, is maintained, but there's a wear and tear that eventually can catch up with the body and brain in terms of diseases, and the diseases of modern life are examples of this. The other part of this is that when we think about a mediator like cortisol, we have to realize that acutely it actually enhances adaptive immune responses. It promotes memory for things that we want to avoid that are dangerous. It's involved in energy replenishment and cardiovascular function. But chronically, when it's overproduced and not turned off or not turned on when needed, then it can be involved in suppressing immunity, impairing memory, promoting bone mineral loss, muscle wasting, and the metabolic, participating in the metabolic syndrome. And one of the most often overlooked aspects of this is that we have a normal circadian rhythm of cortisol. The elevation in the morning before we open our eyes is very important for getting us active, increasing our hunger for our breakfast. And as has been shown, there is an ultradian pulsatility along with this circadian rhythm. And if this doesn't occur, various consequences occur, including systemically promoting obesity and diabetes, and centrally work by Dr. Connor Liston, who is a former MD PhD student now on the faculty of Cornell Medical School. In his postdoctoral work at NYU, he showed that you could see on the slide on the left the formation or the existence of spines where synaptic connections are made on neurons in many parts of the brain. This happens to be in the cerebral cortex. And you can see arrows that point to the formation over a 24-hour period of new spines and the disappearance of others. So there are these synaptic connections that are continually turning over, not all of them by any means, but enough of them. And when he disrupted the cortisol secretion, turned it off chemically, this turnover stopped. Moreover, when he overloaded the system at wrong times of day for cortisol when it should be normally low, or disrupted this ultradian pattern, not only was this disrupt this turnover of synapses, but it also impaired motor learning and possibly other aspects. So our normal lifestyle often plays with our circadian rhythm, our sleep-wake cycle, and it does have consequences for how our brains operate optimally, and we are often our own worst enemies in terms of how we handle this. Now, there's more than cortisol. Cortisol is one of many mediators, along with pro- and anti-inflammatory cytokines, the sympathetic and parasympathetic nervous systems, a little bit of oxidative stress, which is necessary for survival. Too much is damaging. Dihydroepiandrosterone is a steroid that's also very important as kind of a counteractor for cortisol. These all talk to each other. That's what the arrows are about. And they operate in a nonlinear way, so when one goes up, often others change in their levels as well. Moreover, as the slide shows, they are simultaneously affecting organ systems throughout the body. So one of the consequences of this, of course, is that for many disorders, there is what we call multi-morbidity. For example, for people with mental health problems, the minority express, say, only one disease, say major depression or something like that. For the most part, most people will show one other, two other, or multiple other diseases. This is not surprising since these mediators are working throughout the body, and often then a person has to be treated. If they're treated aggressively for one disorder, this may disrupt other disorders. So keeping in mind the balance of the body is very important. Now, back to the brain. We've heard the brain shows a considerable, surprising amount of plasticity, even in adulthood. And I want to give you just a little background on this from some of our own work over the years. We know, in general, from our and other work, that their dendrites of nerve cells can either grow, become more branched, or become debranched, less branched, that there is this turnover I already mentioned of a number, not all, but some spine synapses, and that there's a limited amount of neurogenesis, especially in the hippocampal formation, a part of the hippocampus called the dentate gyrus. And these are altered by experience, and circulating hormones and endogenous chemicals are involved in mediating all of these structural changes. Some time ago, we discovered that the hippocampus has receptors for cortisol, for glucocorticoids, and this was the first time that anyone really had looked beyond the hypothalamus to realize that the brain as a whole, which we know now, is responsive to stress hormones. It's also responsive to sex hormones. Perhaps we could say a little bit more about that later on. And there are important sex differences throughout the brain based upon that. Glucocorticoids, or cortisol, has biphasic effects on memory and neuron excitability. The inverted U is an important concept in all of this stress physiology because a little bit is good for you, too much can be too much. The basic idea behind allostatic load. The hippocampus is important for mood regulation, and it's a vulnerable brain structure for wear and tear over the lifetime related to aging and also, as many of you know, to dementia. Repeated stress in a young, healthy adult animal causes the apical dendrites of neurons in the CA3 region, which is a critical region of the hippocampus, to become shorter and less branched. But the basal dendrites don't change. And moreover, when the stress ends in a resilient, healthy animal, the dendrites grow back. But they don't always grow back exactly as they were before, which means that the brain is, as I said, not the same, not really showing a reversibility. The neurogenesis occurs in the inner part of the dentate gyrus. With repeated stress, there's a reduction in neurogenesis, and with a chronic repeated stress, you can even shrink the the volume of the hippocampus, especially the dentate gyrus. But regular physical activity, as I'll show you momentarily, has the opposite effect. In translating to the human brain, researchers have found that the hippocampus becomes smaller in major depression, in type 2 diabetes, in post-traumatic stress disorder, in Cushing's disease, where there is some resilience when the hypercortisolism is corrected. But you don't have a disorder because a history of chronic stress, chronic jet lag, lack of exercise, and a general state of chronic inflammation have all been associated with a smaller hippocampus. The good news, however, is that regular exercise can enlarge the hippocampus. Intense learning in a study on medical students in Germany was shown to increase the volume of the hippocampus and sustain it for a period. And antidepressant treatments, of which exercise is one of the best for the more dysthymic or milder forms of depression, is also effective. There have been a few studies with chemical antidepressants showing the same thing. The poster child of behavioral interventions that can affect the brain is this study from Kirk Erickson and Art Kramer and colleagues at the University of Illinois, showing that getting elderly couch potatoes off their duffs to walk 60 minutes a day, 5 out of 7 days a week, over 6 months to a year, actually enlarged the volume of both the left and the right hippocampus without major changes in other parts of the brain that they looked at. So exercise, physical activity, as we all, I think, are realizing, is one of the best things that we can do to maintain not only body health, but also brain health. Now, the hippocampus is one important brain structure which is involved not only in memory and mood regulation, but also in shutting off the physiologic stress response. The amygdala is another important brain area involved in fear and aggression, and we've heard a little bit about that already. It turns on stress hormones and increases heart rate. And the prefrontal cortex, we've also heard about, is very important for decision-making and self-regulatory behaviors, but is also important for shutting off both the autonomic and the neuroendocrine stress response. And what is particularly important to note is that the normal response to chronic stress, and we're not talking about anything close to brain damage, while it causes dendrites in the hippocampus to shrink, suppresses neurogenesis, it also causes neurons in the prefrontal cortex to shrink. And at the same time, it causes neurons in the basolateral amygdala, which is involved in anxiety and fear, and not shown here in the orbital frontal cortex, which is involved in vigilance. These dendrites expand. So in a dangerous situation, the brain is programmed to be more responsive to possible future stimuli, more vigilant, and yet when the danger passes, if these changes don't show some recovery, then the individual or the animal has an anxiety disorder and it has to be treated by some external means. So the brain gets stuck. So reversibility and resilience, reversibility doesn't occur, but resilience and recovery are important characteristics of a healthy brain. Our colleague Shona Chatterjee in Bangalore, India, has been a pioneer showing these changes in the expansion of dendrites in the basolateral amygdala related to chronic anxiety and as a result of chronic stress. We did an experiment together in which an acute chronic stress, putting an animal into a severe restrainer for the first time in its life, caused over 10 days an increase in spine synaptic density in the basolateral amygdala, caused a delayed increase in anxiety. And we discovered by accident that either a modest stress before the animal went in that restrainer or giving a systemic elevating corticosterone prevented this whole effect, a PTSD-like effect. And there are several studies showing that elevating cort during or right after traumatic stressors, like a car accident, will actually reduce the incidence of PTSD symptoms. So this is another example of why cortisol is actually not a bad guy, but really is important for normal brain functioning. John Morrison, who used to be here at Mount Sinai, now in UC Davis, we collaborated with him over quite a few years, and he is in his lab really. Our collaboration showed that stress causes these dendrites in the medial prefrontal cortex to shrink, the orbital frontal cortex to grow, and in the same Connor Liston, in a study on rats, showed this, but in a study at Cornell on medical students going through their year using the perceived stress scale and using functional imaging, found that high levels of perceived stress were associated with reduced cognitive flexibility using cognitive tests while they were in the scanner, and reduced functional connectivity involving the prefrontal cortex. This notes that the good news is that these effects were reversible or recoverable after a vacation, and so at least the young brain has some capacity for resilience. Other studies that the Morrison lab did, often in collaboration with us, showed that there is an impairment of this plasticity or of resilience with aging, also with certain kinds of persistent chronic stress. So the brain does have that capacity even as it ages, which can perhaps be restored by increasing physical activity and changing some of the other factors in their lifestyle. Now, the role of early life stress involves, of course, produces individual differences that are related to not only our genes, but also our developmental history and our experiences, as we've already heard, as we well know. And we're certainly aware of the fact that not only adverse childhood experiences, but also chaos in the home leads, among other things, to impaired development of the prefrontal cortex, altered wiring of the brain, as we've also heard, as well as increases in incidence of disorders such as depression, substance abuse, antisocial behavior, cardiovascular disease, diabetes, obesity, and so forth. One of the important additions to this is this connection between metabolic dysregulation, insulin resistance, prediabetes or diabetes, and the occurrence of depression and the increased risk for dementia. We're involved in a study with, among others, Dr. Natalie Razgon from Stanford on this using animal models and beginning to obtain data on people with humans with depression. But just to go back to the brain areas that we're talking about, we know from animal model studies and from studies on the human brain that the hippocampus is smaller in people growing up in poverty, in people with who develop low self-esteem, perhaps because of early abuse and neglect, and this is associated with an increased risk for PTSD. The amygdala is larger and more active in depression and anxiety disorders, and it's actually, as Sonia Lupien in Montreal has shown, it's larger in children living for some long period with a depressed mother. And the prefrontal cortex, of course, has, as I've already mentioned, has this important role in self-regulatory behaviors, and the complex connectivity of the prefrontal cortex with other brain areas is altered, so that there is a tendency for impaired self-regulation, which can last for a long period of time, even through lifetime if without intervention. And so, using this nice figure from a wonderful review article by Neil Halfon, talking about factors in early life that can either impair this trajectory to lead to poor health, physical and mental health, or elevate it in a more positive direction. We should consider some of the interventions that have already been used, and maybe think about others that could be developed. And I know some of this will already be discussed here, has already been mentioned. The nurse-family partnership, well known for its effects in dealing with an expectant mother and helping her and her partner transition as the child is born, providing emotional and educational support, has been very, very effective, as documented in papers by David Oles and colleagues. A recent remarkable study in JAMA Pediatrics by Gene Brody and colleagues in Georgia did a 7-week intensive intervention with adolescents at age 11 and their caregivers, and followed them out through age 18 and 25. And this report, which obviously caught my attention because of its effect in the volume of the hippocampus, is that for the control group, the number of years in poverty was associated with a progressively smaller hippocampal volume. And yet this intervention showed a complete protection of that aspect of the effect, along with some very impressive effects on mood-related depressive symptoms. And I think other data is coming out from this study, which suggests that that adolescence is a very important window of opportunity for intervention, and that we don't have to do everything early in life. It isn't over, even if some bad things have happened during that period of time. Generally speaking, then we have physical activity, mindfulness-based stress reduction, social support and integration, and meaning and purpose in life. And the last slide from work by Tanya Singer, showing in young adults, well, between 20 and 55, the use of various kinds of mindfulness interventions actually caused sculpting the areas of the cerebral cortex in significant ways, along with improved behavioral outcomes. So looking to the future, plasticity opportunities to use this, I think, is one of the important directions, and a large group of people to thank, including the National Scientific Council on the Developing Child and the Hope for Depression Research Foundation. Thank you very much.